introductory remarks will be very brief, so I apologize if I need to say more, but I don't have time. Glad you're here. This is our second in a series of lessons designed to teach us the evidence of the God we serve in the natural world. I'm thrilled to be a part of this, to share the things that I have learned through the years that I pray will help you as well. Thank you for joining us, and I ask you to give me your close attention. I will remind you, if you were here last night, and tell you if you weren't, I don't allow sleeping in my class. And you are in a class. I welcome you tonight to Biochemistry 101. If you haven't had any of that, it's okay. I'm going to teach you what you need to know to make the points tonight. The other thing I want to say is this material is a bit heavy for folks who are not chemistry and math folks, and I make no apology for that. You don't have to remember all this. There will be no tests. What I want you to be is overwhelmed with the majesty of God. And I can't do that properly without some detail. So please stay with me best you can. And I hope by the time we finish, you will be thankful more than ever for the God we serve. So, my little clicker's not here, so down below, there it is. April the 8th, 1966. Raise your hand if you were alive then. Make me feel better. Time magazine. Is God dead? There was a reason the magazine featured that right then. It was a heady time in the scientific world. So let me give you a little background. You lived, class, at a time when mankind has learned more about the chemistry of living things than in all the history of mankind prior to this. In the last 75 years, we have opened up whole new vistas about living things and how they work. You've lived through it. It's an exciting time. Kids, it's an exciting time to be interested in science. And one of the things I always like for children to leave thinking, I want to be a scientist and learn more about God's world. You don't need to be afraid of science. If you study deeply and keep your mind open, you'll believe in God more than ever. Get finished. In the 1950s, many things broke loose. It was during that time that James Watson and Francis Crick discovered the structure of DNA. Only changed everything. And the whole direction of study about living things has changed since then. And they won the Nobel Prize for that sometime later. What you probably don't know is Frederick Sanger also mapped the first protein in the 1950s. Insulin. He actually mapped insulin, showed what it was made of. Amazing breakthrough, for which he won the Nobel Prize. And Frederick Sanger is one of the few men in history in the sciences that won 2 Nobel Prizes. But some years later, in the 60s, he was the first one to map the structure of DNA and show how it was laid out. For which he won a Nobel Prize. All of this unfolding as people began to say, and by the way, you probably remember that Darwin published On the Origin of Species in 1859. So in 1959 was the century celebration of Darwin's contribution. And you can be assured that all over this country and around the world, he was being glorified as one of the greatest in the history of mankind. And his theory was being broadly proclaimed as one that shows you don't need God to bring about species of life. All of these things were unfolding in the 50s and 60s, and I was in high school and college in those days. And I can tell you that it was a time when scientists were saying, many of them I had experienced, if you're a believer in God, you better get over it. Because the way things are going, mankind is learning more and more, and the more we learn about how things work in the natural world, the less we need God. And so, is God dead? Was a practical question in the 60s, coming out of what was all happening in the science. A couple more things just for interest. And this is a class, so you're allowed to answer my questions. I'm not going to pick on individuals. Do you remember, some of you, what happened in the 1950s that scared us to death in America? It had to do with space. Thank you. You children don't remember because you weren't alive. The Russians put an object up in space for the first time in history. They called it Sputnik. And it scared us all so badly because we knew they were going to take over space and rule the world. So what happened is our government decided to emphasize science education. And all of the curricula that were used in high schools and colleges were rewritten. And if I may just throw in a little aside there, I was privileged to get in on some of that. Because I was a chemistry education major. And I was privileged to get a scholarship to Harvard University on that very basis. Because the government gave Harvard enough money to give 50 scholarships. People all over this country. I got the one from Florida. And the purpose was to go study at Harvard to be a better science teacher. So I hope you think I'm a better science teacher tonight. But one of the things I took part in was rewriting the chemistry education for high school called Chem Study. Some of you may remember that. And I also had a part in some of the new math, your favorite thing. But all that was unfolding in the 50s and 60s as the excitement was rising for what science was going to do for us and tell us. And one of the things that I was told by some of my professors was that it's telling us you don't need God to comfort them. So, April the 8th, 1966. I'm starting with that for a reason. Because you see this up here? A.W. Dicus was the academic dean at Florida College, where I happened to go to school first. He was a physicist and widely respected scientist. He wrote the song you just sang. Our God is alive. And thank you for being so lively with that singing. Because what you said is, our God, he is alive. In him we live and we survive. The third verse of that song says, secure is life from mortal mind. God holds the germ within his hand. Though men may search, they cannot find. For God alone is understanding. We had a biology professor at Florida College who refused to sing that verse. And the reason he did was because he didn't think it accurately reflected the way things were going to be. He truly believed that over time we would have understood how living things came about and learned how to do it ourselves and learned that it happened without God. I wish he were still alive today. Because, folks, after another 60 years since those days, we know less today about how life came from non-life than they thought they knew then. In fact, the more we have learned about the complexity and the amazing things about living things at the chemical level, the less we know how that could possibly have come about without a designer. And that's part of what I want to tell you about tonight. It's a challenging topic, and I'm ready to tackle it. Thank you for singing that song so enthusiastically. In this text and in his book, Radical Theology and the Death of God, Altizer and Hamilton said the following: we must realize that the death of God is an historical event, that God has died in our cosmos and in our history, and the dead God is the God of the historic Christian church and beyond the church of Christendom at large. These are preachers, ladies and gentlemen, theologians who were explaining to the world in this text and in that book, God has died in our time. How's that possible? That's only possible, class, if we made him up in the first place. But that was the notion in that day by many preachers, not just science teachers. So you've also lived in the time as science is blossoming, when science itself has begun declaring you don't need God. And science, there's no way, folks, that science can do that. There's a limit to what science can do. It cannot answer the most significant questions that we have to discover. So I kind of like this new book. Is Atheism Dead? By Eric Metaxas. I'd highly recommend it to you. I mentioned last night there's a bibliography in the back, and they have a copy for you if you want it. It has all these books in it. Some of them are heavier than others. This one it's written by a non-scientist. Eric Metaxas is not a science guy, but he's a good writer, and he does his research. It's well done. And he says in this book, since the Time article in 1966, roughly 5 things have arisen to challenge, and I will argue to overturn, the secular consensus that formed in the wake of that article. That's an important statement, because it is absolutely correct that after that Time article about is God dead, there was a secular consensus, a movement that we don't need God. And what Metaxas is saying, and others I will quote tonight, is that since those days, we have learned things we never knew before that overthrow the secular consensus. So here are the 5 that he quotes in his book. There's a beginning, class. Did I introduce that last night? Yes. Science is now speaking that matter, space, and time has a beginning, and the evidence for that from science is overwhelming. And the great majority of scientists in our modern time, even those who are atheists and agnostic, have to admit that's the case. That's not my theme for this particular series, but it is a fact. Second, fine-tuning. Did we talk about that last night, class? Fine-tuning, how things have to be. You say it with me, class. Yeah. Lots of things have to be just right or you wouldn't be here. That's a fact. And in the last 70 years, we've discovered hundreds of things in the cosmos and in our solar system and on Earth that all have to be, say it again, just right or you wouldn't be here. So that's a powerful thing we've discovered. The third thing is stupefying complexity. I love that word. I want you stupefied when you walk out of here. Stupefying complexity. That's our theme tonight. The fourth thing he mentioned is archaeological discoveries, which relate more to the truth of this book. And that's not our theme tonight. And the final one he says is we've learned what atheism is and what it produces. And just for time's sake, I'm not going to go there tonight, but you need to read his chapter on atheism. The 20th century, class, was the age of atheism. It was the century when governments were atheistic. You think about communism, about Hitler and his horrible regime. You think about so many communist regimes in Cambodia and Vietnam that killed millions. You want to know what atheism has to offer? You go read about the history of communist dictatorships in the 20th century. We don't need atheism for all kinds of reasons. But all of those things, he would say, would argue that atheism needs to be dead. I say amen to that. So, tonight, as we did last night, because we're having this series in a church building, in which most of you are Bible believers and believers in God already, I think it's fitting to start this discussion from the Bible So we're going to look again at Romans 1, verse 20, which is the theme passage from the scriptures that says, "For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse." That passage teaches in the Bible that you can come to believe there's God without the Bible. You listening to me? You don't have to have the Bible to come to be a believer in God. The natural world can convince you of that. That's part of why I'm here. And I like to call that God's other book, as did many of the great scientists of the old days. They said, we're just studying God's other book. And from that book, you ought to be at least able to know that he's eternally powerful and he's not part of nature. He's divine. You can learn that without this book, class. So please don't get upset if for the rest of my time tonight, I'm not using this book. Look, this is the Word of God. I'm convinced, and I preach from it all the time. But not now. I want to turn with you to God's other book, which you haven't studied deeply enough. And I want to help you see some things maybe you've never seen. It's called the argument from design. He says, by the things that are made, you can conclude there was a great maker, powerful, and not part of this world. I'm convinced of that. So here we go. Are you ready for a chemistry lesson? Please say yes. Yes. Thank you. Welcome to Biochemistry 101. Components of life. What we're studying is life. Ladies and gentlemen, life is chemistry. And chemistry makes up life. Don't mean that's all there is to it. But in terms of the physical world, chemistry explains living things. So I thought to begin, we ought to read from one of those great Nobel Prize winners. His name is Francis Crick. He wrote a little book in the 1980s called Life Itself, Its Origin and Nature. If there's anybody that will be able to tell us about life and what it's like, it's Francis Crick, Nobel Prize laureate. So I begin on page 72 of this little book, and we'll look a little bit about the critical components of life. Life as we know it on Earth appears as a synthesis of two macromolecular systems. And I've lost you already, right? Every living thing, class, every living thing has two giant types of molecules. Macro means big, right? Two types. The proteins, because of their versatility and chemical reactivity, do all the work, but are unable to replicate themselves in any simple way. Very important point. Proteins are the workhorses. And in every living thing, from a single-celled organism all the way up to you, there are thousands upon thousands of proteins doing work all the time. But they can't reproduce themselves. The nucleic acids, so let's walk through this here. The structural materials are proteins, and they make up the shape and mobility of living things. So, they're assembled from amino acids, which we'll talk about in a moment, but they make up the structure. Secondly, there are the tools and machinery. That's the enzymes. An enzyme, though, is just a special form of a protein. So both 1 and 2 up here are proteins. Just so you get in practice, I want you to say that word for me. Both 1 and 2 up here are what? Proteins. Proteins, good. Class, that is the answer to most of my questions. So you're going to be ready to say that word tonight instead of just right. Okay? Finally, the blueprints. The genes are nucleic acids. The DNA and the RNA are for reproduction. So here's what he says. The nucleic acids seem tailor-made for reproduction, but can achieve rather little else compared to the more elaborate and better equipped proteins. And he says this, please don't take offense, ladies. RNA and DNA are the dumb blondes of the biomolecular world. Fit mainly for reproduction and can't do really demanding work. Sorry, that is him, not me. That's probably the one thing you got to remember. Yeah, horrible. Whoa, that was rough. What you need to remember is these guys right here take care of reproduction. They contain the information and direct reproduction. The proteins do most all the work. By the way, I mentioned last night, I usually use a laser pointer. But you can't point a laser at a TV screen and get anything off. So I have the old-fashioned. We'll make it do. All right. So, introduction to living things. We're going to keep it as simple as we can. Proteins and nucleic acids. The problem with living things is you have to have a both. And he goes on to say that here. The problem of the origin of life would be a great deal easier to approach if there were only one family of macromolecules capable of doing both jobs. But life as we know it always employs two. Class. You can't be alive without proteins and nucleic acids. Is that clear? That's on the pop quiz in the morning. Joking, of course. But if you were in my class, you can be assured it would be on the quiz in the morning when we meet at 8:30. Now, you noticed in that chart that it says that proteins are made up of amino acids. So I have before you on the screen, class, your introduction to amino acids. In every living thing, whether it's a single-celled organism or you, your proteins are made up of these. Now, let me interrupt myself there a minute. What do you think a believer in evolution would say about that? Here's what they would say. Why is it that all living things have a common chemistry? It's because we all came from the same common source. Way back millions of years ago, we started as single-celled organisms and evolved into all the places we are now, and that's why we have a common chemistry, because we have a common ancestry. That, class, is not a stupid answer. It's a reasonable answer. So what do I say about that as a believer in God? I say it's just as reasonable for a creator who is going to make a system of living things that interplay with each other to have a common chemistry. You don't have to start over every time you make something, especially if your plan is for it to work together in a system. So you cannot answer that question with just this data, class. Both of those answers are reasonable. So we must go much further than this. But it is a fact, class, with a few little exceptions. There's proteins in every living thing made up of those 20. So your first task in biochemistry is to memorize it. Now start easy. I just want the names and the abbreviations, which is mostly the first three letters. So let's read them here. Glycine, G-L-Y, alanine, A-L-A, valine, V-A-L, and so on. Arginine, cysteine, methionine, histidine, the 20 amino acids. The reason you need to learn those is because they are only the key to all proteins. And proteins are the key to life, as are nucleic acids. That's very important. In addition to that, if you were in my class, the next week or two, we would learn the chemical structures and why they are so important. Because depending on the amino acid, depends on, well, how that protein is going to react and the sequence of it. So it's important. Now, I'm not going to do all of that tonight. I will point out a couple of things, though. They're called amino acids because every one of them has a nitrogen and hydrogen group. It's called an amine. So they're called amino acids. You got that? They also, each one of them have a COO group. That's a carbon and 2 oxygens. That's the part that produces an organic acid. So it's called an amino acid. Every one of them. So they all have things in common, but they're all different because the rest of their chemistry is different. All right? That's not too difficult. I'm laying the groundwork for where I'm going, class. And if you don't have the groundwork, you're lost. 20 amino acids. All right. Every protein is a combination of those 20. It's very much like an alphabet. In English, we have 26 letters. Every word in English is some combination of those 26 letters, isn't it? Even anti-disestablishmentarianism. It's some combination of 26 letters in a string, and some of them mean something, don't they? Certain strings of letters mean something. The same is true in protein language. Certain strings of amino acids mean something. They mean a specific protein that does a specific task. All right? I apologize to every protein that we haven't spent 3 weeks on this, but I have to go on. The other macromolecular system is the DNA and the RNA, and we have to spend a little time on that. So here's the structure that Crick and Watson discovered. It's double helix. Can you see there's a strand here wrapped around this strand, kind of wrapped around each other? And then there are cross pieces. Do you notice on that chart, class, that there's exactly 4 colors on those cross pieces? Because of that, it has a capacity to reproduce itself, which allows you to reproduce. It contains a language with 4 letters, A, T, G, and C. That stands for adenine, thymine, guanine, and cytosine. That's question 2 tomorrow. And those 4 bases, which form the cross pieces in the DNA, are critically important because they form, class, a language with 4 letters. And that language we've learned to read. We know exactly what it's saying. It only has 4 letters. So those 4 letters are critical. And believe me, if you were in my class, we would spend a whole week just learning about those 4 substances. For now, you just need to know that makes up the language in living things. And here, class, is a brief summary of the language itself. We call it the genetic code. You'll notice that on one side of that chart, the letters thymine, cytosine, adenine, and guanine. Across the top, you see them again, and down this side, you see them 4 times. And the reason it's laid out that way, class, is because by that method, you can take those 4 letters and form every 3-letter word that's possible. Now, if you were in my math class, I'd ask you to do that arithmetic. How can you know how many words you can make 3 letters at a time where you can repeat letters and you only have 4 letters to use? That's a combination problem. And there's a math formula for that. So I'm going to tell you what it is. If you have 3 places and you can put 4 things in each one of them, it's 4 times 4 times 4. So do the arithmetic, kids. What's 4 times 4, kids? 16, and 4 times 16 is 64. Thank you. There are exactly 64 words up here, class. So let me help you learn to read. Everybody's eyes up here. If the first letter in the word is T, the second letter is T, and the third letter is T, that's this one right here. Can you see it? That word is pronounced This is not a spoken language. Right next to it in parentheses is a three-letter word abbreviation, PHE. So what it's telling you is, means feet. That's very helpful, isn't it? If you had memorized the chart I asked you to memorize, you would know that PHE is phenylalanine, which is one of the 20 amino acids that you just learned. So class, what I've just shown you is that particular code codes for a specific amino acid. It means phenylalanine. You see any others up there for PHE? Right under it, TTC also means PHE. See any others? I think that's it for phenylalanine. There are some repeats in here, class. There are 64 three-letter words, and there's only 20 amino acids, right? I call that a backup system. You have more than one code for amino acids. Like leucine's got a bunch of them. There's 6 of them for leucine. Try another one just for fun. How about ACT, ACT? That's THR. That's threonine, class, threonine. One of the 20 amino acids. Okay? Now there's some exceptions here. How about this one right here? TAA means term. Anybody want to make a guess? That's not one of the 20 amino acids. So what does term mean? Terminate, which means stop. So that code means stop. And there's another one, right? TAG means stop. I think that's it, isn't it? Oh, TGA means stop. So there's 3 codes for stop. What you don't see up here, there's this one, ATG, for methionine, also means start. So let me summarize, class. What you have in this code is a series of 3-letter words that stand for the 20 amino acids and stop and start. And you say, so what? So what? It only means everything in how living things function. Because it's this language, ladies and gentlemen, that directs your body and how to build proteins out of amino acids. And it's a rather complex process that we've uncovered in the last 75 years. But it's utterly stupefying. So my job for the next few minutes is to stupefy. I'll start this way. Ladies and gentlemen, if you were a jury and I was trying to convince you there has to be a creator behind this, the first thing I'd start with is every living thing had to be created because built into it is a language. Where do languages come from? Can you tell me, class, any example in the history of mankind where a language came without some intelligent source? You cannot. Languages are produced by intelligent sources. Every living cell has a language in it. And not only that, it's being used at a phenomenal rate of speed to do things based on the language. That's fact number 1, ladies and gentlemen of the jury, that should say to you, that was made. It did not come about by natural causes. But there's so much more than that to talk about here. So all 20 amino acids are coded here, plus stop and start. I had him draw this picture for me, or pull it out. Matthew did this for me today. You heard of M.C. Escher? He's a great artist from Holland. He's known as the artist of almost infinity, because he has this mastery of drawing things that all go together and everything depends on everything else, and it looks like there's no stopping it. Look at this. This is his drawing called Drawing Hands. So you start over here. This hand is drawing that one, isn't it? I mean, he's drawing it here. And it's two-dimensional to start with on a piece of paper, and then it grows into a three-dimensional thing over here, and then that hand is drawing this one. As it starts in the same fashion, and it constructs a three-dimensional figure. But all of that placed together, can you have one hand without the other in this picture? No, he constructed it in such a way they're absolutely interdependent. Everything about it works together. How do you get something like that, class? You get it from a master artist like M.C. Escher, and no other way. That was produced by intelligence. Would you say? So what I'm about to present to you is that exact same notion. All of this functioning I'm about to show you is completely interdependent. In other words, you have to have it all or it doesn't function. It has to work together in concert or nothing happens. Much like you don't have this without the artist. It all has to play together. In the picture, you have to have it all to get the picture. So I thought that was a good introduction. What did I do with my thing? There it is. All right. There's a whole lot more to this story than just the language, so watch. You see up here, this is the nucleus of a eukaryote cell, and in it it has the DNA string. You see it here, this strand, interwoven strand. So the information in the DNA is tied up here. Out of that information, we're now going to produce proteins. In order to do that, you first of all have to transcribe the information. It's tied up in the DNA. But if it just stayed in the DNA, you're never going to have any proteins or anything alive either. Second, it has to migrate outside the nucleus. And thirdly, it has to be translated in this ribosome down here, which looks like two ducks hugging each other. Which it doesn't really look like that. That's just a picture. And then out of that ribosome comes the constructed protein made up of a whole series of amino acids. There's a lot to this story, class. But it is all absolutely interdependent. So I told you the answer to my questions, right? Just a moment. Up here, when you're going to transcribe the information off the DNA, what do you think, class, it requires for the DNA to be able to make copies of itself? Protein. Very good, only that was weak. So I will say it again. What do you think it takes, class, to be able to copy the information off of the DNA? Protein. Yeah. It's not good enough just to have DNA. You have to have protein. Once the information is copied and you've made messenger RNA, ribosomal RNA, and transfer RNA, which we're not going to have time to talk about all that. Once that's done, then the messenger RNA has to be matured. Do you see the little white regions here? Those are called introns. They're snipped out and reorganized and edited, so that's called the maturing process. What do you think it takes to make that happen? Good. I think I won't ask somebody to keep track of how many times we say proteins. But just keep that in mind. And by the way, it's not just one protein up here, as you'll see in a minute. Nor is it just one protein here. It's a lot. Now we've got the mature messenger RNA and the other RNA units, and this is going to migrate now outside that little hole right there. The nucleus has holes in it. And no respectable cell is going to let just anything go through those holes. So what do you think, class, guards the gate here, that hole that allows only certain things to go through? Protein. Yeah. there's a protein that does this. Come on in. But only for certain things. And by the way, I'll mention it here in case I forget later. If that messenger RNA that's trying to come through that hole isn't the way it's supposed to be, it won't open. In other words, if something messed up along the way, this is part of the quality control system in your body that's everywhere. You won't let it out of that hole because you don't want a messed up messenger RNA going out there in your cytoplasm. Once it gets outside, it has to be picked up here by the ribosome. The ribosome class is made up of protein and RNA. Lots of proteins. And it picks up that messenger RNA and reads it through it like a tape through a tape player. And it reads that 3-letter code. You see it? When it reads the code, it sends out a signal to the transfer RNA to bring in the appropriate amino acid right up here and bring it into the ribosome so it can be added to the string which you're making, which is the proteins. Class, guess what it takes to help the transfer RNA bring the appropriate amino acid up to the ribosome? What do you think? Proteins. Exactly one protein for all 20 amino acids, so that's 20 more proteins. In addition to that, once this brings it up here, you have to have another, what do you think, to help it do the job inside here that is very complex to allow the information to be copied from here onto that copy so you know we're matching correctly so you can then build the right protein. Guess what it takes for that? And then finally the protein is built and released, and I've skipped several things, class. Once it's constructed, you're not finished. Because the protein not only has to have a primary structure, which is the string of amino acids. By the way, may I say it? A string of amino acids is a protein. It's not good enough just to have a string of amino acids. It has a secondary and a tertiary structure that go with it. It has to fold up in a certain way or it doesn't work. Guess what it takes to help it fold up properly? Protein. Thank you very much. So every step of this process requires the information from the DNA, and it requires proteins every step of the way. And class, listen to me closely if you don't remember anything else from tonight. Starting with the very first protein right up there at the top, that protein that is needed to build every protein also has to be built. You listening? So how did this whole thing get started? Because to build even one protein, you need about 50 other proteins already there, each one of which had to be built. Are you getting me? It's a massive problem for something that came about by natural causes, by random mutations acting on random matter over millions of years. That's a problem. So that's a little blow up that first stage. What I'm going to do now is I'm going to skip this part and go to a video to make it really come alive to you. Don't you forget it. This entire process requires an intermingling of the DNA, the RNA, and the protein. And we're now going to talk to you about a video produced by this gentleman. His name is Drew Berry. He works at the Institute of Medical Research in Melbourne, Australia. He's an amazing biomedical animator. He likes to make living things on a small scale come alive. So if you want to have some fun, children and adults, go look at his website. He's made a bunch of amazing videos to show you how things work. And we have picked one tonight to share with you. All right, what you see here before you is the video clip that Drew Berry made to show you how proteins are built. And this is supposed to be active enough that it's real, like it's real. But you know it's a video. It's an animation. So watch. This is the DNA strand as he portrayed it. This is a glob of, what do you think? Right. Yes, thank you so much. Do you notice the colors? They're not that color class, neither is the DNA. He put the colors in to make it pretty. Right? But it does illustrate there's 1, 2, 3, 4, 5, 6 different proteins right there. It's a glob of protein that right along the DNA strand that allow things to happen. Okay, so proteins are good. All right, now his job is to stop and start when if he messes up, it's not good. But he did a good job yesterday, so I'm counting on you. So here we go. We're going to ride a little bit here, and then I'm going to stop him. Let's go. Go. All right, so this is the start of a gene for a bone strength protein. That's what we're going to build here next. Stop right there. Good job. You see this right here? That's vitamin D. Critical component to building proteins. So get out in the sun. Get your vitamin D class. You need it to build proteins. As you will see here as we proceed. Let's go and watch the vitamin D. It's going to join up to that protein. Stop right there. And it turns it green. No, it does not. It does that so you'll know something's changed. And green means go. That's why I did it, I guess. But that protein has now been activated. The vitamin D activated, and it says now it's time to get to work to start build this bone strength protein. Okay, let's go. Here comes more. What do you think? Yes, stop. I think we lost some of them. There's 1, 2, 3 more proteins in there that have been added now to the glob of proteins to do the specific job. Class, if you were really in my biochem class, we'd stop right here and I'd spend the next 2 or 3 days telling you what those proteins are doing. And it's a lot. This man thinks that life is just riding along having a happy time. No, there's a lot going on there. And may I add right here, I was just reading today, right here, in this book, The Cell's Design by Fazeli Rana. What all's going on? And I have to tell you, class, I was sitting there in my room, and I started crying. Just because of the amazing things I was reading. I can't get all that across to you. It's too much. I'll tell you what happens right here. In this process right here, there is not only work going on to allow you to copy the information, there's work going on to make corrections if it makes a mistake. You listening? There's a system that helps correct things built in every step of the way. Folks, that kind of thing does not without intelligent, careful consideration. Amen. So that's happening all as we speak here, as we go along. So let's go. When the time is right, stop right there. Ribosome, ribonuclease splits off and rides along down here. And these guys have already done their function. And so they're now left to go back into the cytoplasm and be reused. It's recycled. And we're going to ride along here with the ribonuclease, which is another, what do you think? By the way, any word that ends in ASE is an enzyme, which is a protein. So if it says ACE, it's a protein, an enzyme. All right, so let's ride. Here we go. You can see now, we're making the transcription. And we're going to look down inside here in a minute. Stop right there just a second. Here's the DNA moving along this way. The RNA is coming out here. This is the copy that's being made. And this is the blob of what, class? Protein. Riding along here that allows the copying to be done. So let's keep going. We're going to magnify this now. Look inside. See all the stuff around the outside? That's all the materials that are needed to build this messenger RNA. Got to have it. And it comes migrating inside here. There, stop right there for just a second. We now magnified this, and what's happening down inside here is a lot. You see all this stuff coming in here? It needed to go in to be added because we're making the messenger RNA piece by piece down inside. And then what's happening in here is the DNA, which wraps around itself, has to be unzipped. So let's do it. Down inside. Stop. You see it unzipped right there? Class, what do you think it takes to unzip DNA? Good job. How many do you think? Well, I don't want to ask you that. I'm going to tell you that there's several involved. And not only that, as that is being unzipped and zipped back, by the way, what do you think it takes to zip it back up? You ever had trouble zipping things up? That can happen in DNA. It's very important, and it's a complex process that zips it back up without problem. All right, so it's unzipped, and now you have exposed. Let's go a little bit further, Matthew. A little bit further. Stop right there. Can you see the code along here? G, T, A, A, G, C. Folks, those letters are not written there in your cells. They're just identifying what those four things are for you. But they are there. This is guanine, this is adenine, this is thymine. So what happens here? As you unzip this, as an A is exposed, over here you would normally have a thymine, but when you make RNA, you got a uracil instead of a thymine. Don't ask me about that. So that's the case. If it's a G over here, you have to have a C over there. If it's a C over here, you have to have a G over there. It's a mirror image of exactly the information on the DNA. A and T go together, G and C go together, like love and marriage and a horse and carriage. But notice, please, let's go just a little farther, Matthew. Can you see that each one is being built one at a time? One, stop. Now that one was next, and then that one, and then that. Each letter is being copied one at a time. Do you think, class, it is possible that somewhere in your history of cell development, it might make a mistake and shift a little bit? This one? Possible. I'm telling you, there's built into this mechanism a correction mechanism if you have a messed up copy. Right. Do I have time to explain that to you? But believe me. All right. Now, the other thing that this videographer wanted to accomplish was to give you some idea of how rapidly this happens. Did you know that an average protein is 1,000 amino acids long? That means 1,000 three-letter words. That means you've had to make 3,000 copies of letters up here to make one protein. I mean, you could be dead for that finish. No, you couldn't. He's going to show you how rapidly this happens. And what he's shown you so far is pitiful. Let's keep going. Too slow. That's more like it. Stop. That, class, is how fast this is. You're talking microseconds. You wonder why you get tired sitting there listening to me? Your body is building thousands of those every second. While you sit there and do nothing. No, you're not doing nothing. Your mind's working, lots of other things are happening, but your body's busy. This is how it's doing it. Let's go. Wow. Is that incredible? Stop right there just a second. So that all is happening down in here, and we've left out about 600 things, please. But there's a lot of activity going on in there. Meanwhile, the messenger RNA is being constructed. That's a single strand, and it's being released out into the nuclear protoplasm. And once it's finished, it's released. How do you think it knows when it's finished? Because there's a code that says stop. How do you know when to start? There's a code that says start. And you read it, three-letter word, three-letter word, three-letter word, three-letter word, three-letter word, and stop. And you have a string of amino acids, which is a protein. That's the code for the protein. Let's go. Stop right there. The negative about Mr. Drew Berry's videos, he doesn't show you that intermediate step that I showed you, where the maturing takes place. So listen to me, class. Again, I'm sick that I can't spend a day telling you about this. You have to edit that messenger RNA. That takes an editosome, which is a protein. And then you have to put it back together. That's called a spliceosome, which is a protein. Not quite. It's a bunch of proteins built into a big old blob that does an amazing job of splicing and putting back together. And you know what the evolutionists said when we first discovered that that was happening? They said those introns that are being cut out, that's just junk. They're throwing it away. It's junk DNA. And there was a whole series of efforts to say that most of the DNA is just junk. You better be careful what you call junk. Because the truth of the matter is, we now have this little book written, and lots of others that refer to the myth of junk DNA. That DNA they thought was junk is doing all kinds of stuff. There's no junk DNA. I tell you, I got caught by that too, didn't know how to answer. Believe me, young people and older folks, you're going to get questions you don't know how to answer. And there will always be things you will not know how to answer till you die. I'm just going to tell you, you need to wait 20 years and we'll figure it out better. But in the last 10 to 15 years, we've looked at the DNA structure and we've discovered how many different things it's doing we never knew before. There's a whole lot more to this than we've already told you. Now it's migrating. It's come out that hole, and I told you to guard the hole was a protein that is also set up so only certain things go through. What do you think about the other side of that hole? Another protein to guard, get back in. Now our messenger RNA is ready to go. Let's go. And it comes out into the nuclear cytoplasm, and it's picked up by a ribosome. I was reading in this text this morning, they now think that little bacteria probably have 10,000 ribosomes floating around in their little one cell. Very important. Ribosomes are made up of proteins and RNA stuff. There's ribosomal RNA. Okay. What we've got here, class, is here's the messenger RNA going through the ribosome, and it's reading the information on it, which is 3-letter words, right? And each 3-letter word means an amino acid. Then it calls for the amino acid, which these little transfer RNAs here have to bring in. Go ahead now. Here comes the transfer RNA. Here's a big one. Kind of a blow up here. And I'm going to tell you to stop in just a second. Stop. Here's a transfer RNA with an amino acid stuck to the end of it. Class. You think just any old amino acid can stick to that one? What do you think it takes to make that happen? Protein. Specific protein. To get the right amino acid on that particular transfer RNA so you can take it in there and put it in the right place. All right, so the transfer RNAs, there are exactly 20 of them for the 20 amino acids. They have to get the right amino acid stuck to them, and then they have to migrate, and then they have to put it together, which also requires protein. Good, let's go. It's just showing you different ones, you see? Here are the 3-letter words. You see them right here? 3-letter word, 3-letter word, 3-letter word. Coming through here, it's going to read it. Now let's watch inside. We're blowing this up. Here's the transfer RNA. It's bringing and reading the code right here. And up top here, it's adding the appropriate amino acid. Read the code, add the acid. Read the code, add the acid. Except that's way too slow. Let's see if you can speed it up for us. Come on, move it. That's still pretty pitiful. It happened a lot faster than that, folks. But you see the process. Stop right there for just a second. Also built in right here is another safety device. to help pick up a case where something happens and goes wrong. And take that particular protein that's constructed and kill it. You don't want messed up protein floating around in your cells. Keep going. That's the protein. That's hemoglobin being constructed. One amino acid at a time. Stop right there. Did you see that it folded up? The tertiary and the secondary and tertiary folding happens automatically. The quaternary does not. So for most proteins, when that's all done, it still has to go into another vesicle in which inside of it there are more protein to help it fold up one more time into the perfect shape. Because class, an improperly folded protein is a useless protein. It doesn't work. So what you've just beheld is the process that's going every microsecond in every cell in your body, of which you have some 30 trillion, that is building proteins at a phenomenal rate of speed that allow you to survive because they do most all the work that's being done in your body. The chemistry building proteins in every cell is sufficient to proclaim there must be a great designer. Would you say amen to that?
Amen.
Folks, there is no way on God's earth that this happened without a designer, and I didn't tell you 1/10 of the story. But we have learned so much more about the chemistry of living things in the last 50 years that I haven't even touched. You see, what you don't know is that while all of that's going on that I just described to you, there's hundreds of other things happening at the same time and nobody runs into each other. The cell, class, is a self-replicating nanoscale robot. Every cell. Why do I say that? Because it's self-replicating because it can and does reproduce itself entirely. Did you know cells completely rebuild themselves? Not just the DNA. It's nanoscale because its cells are small, down to the level of 1/billionth of a meter. All this stuff's going on. And for those of you who weren't here last night, we have engineers now doing work at the nano level. Yeah, I believe. And they're trying to imitate what they have found in Mother Nature. How about God's nature? And it's a robot because its activities are carried out unconsciously and automatically by precision molecular machines that follow ordinary physical laws, about which we knew essentially nothing until the 1980s and 90s, and mostly since the 2000s. It's no wonder that Anthony Flew, professor of philosophy and atheist, author and debater, for 80 years, class, he was an open and avowed atheist. He debated lots of folks who are creationists. And at 80 years old, he wrote this book. There is a God. How the world's most notorious atheist changed his mind. You need to read that book. And if you have a friend who's an atheist, he needs to read the book. And I'm going to show you one quote from this gentleman who at 80 years old changed his mind about something as significant as this. I hope I'm humble enough at my age, because I happen to have just turned 80 years old, that I could change my mind about something if I'm convinced. He said, I must follow where the evidence leads. And he did. And here's what he said. It has become inordinately difficult even to begin to think about constructing a naturalistic theory of the evolution of that first self-reproducing organism. The enormous complexity by which the results were achieved looks to me like the work of intelligence. It now seems to me that the findings of more than 50 years of DNA research have provided materials for a new and enormously powerful argument to design, an argument that he argued against all his life. But in this instance, he said, I give up. There is no other answer. And he's right. Class, is it reasonable to believe in God in this scientific age? Some would tell you it's less reasonable than ever. I would tell you it's more reasonable than ever. Because the things we've described to you and thousands of others cannot have happened without somebody making it. That's God. I'm going to go home tonight, and I'm going to ask God for forgiveness for the miserable, incomplete job I have done representing his natural world. Because, class, I don't think I stupefied you yet. But tomorrow night, we're going to talk about molecular machines that we've only learned about in the last 10 to 15 years. And we still are learning a lot. And I want to share you just a little bit about what we do know about these amazing machines that are better than anything any human ever constructed. So I hope you don't have to miss tomorrow night. And I thank you for your good, careful attention tonight.